everybody. Can you hear me up there? Can everybody hear me? Excellent. Okay, for those of you who don't know who I am, I'm Sigal Balshine, and it's my great pleasure today to introduce our colloquium speaker, Dr. Amy Newman. Dr. Newman is a professor, a full professor, and also the chair of the biology department at the University of Guelph. And her ecophysiological research is really focused on how environmental and developmental stressors affect small mammals and birds. She's studied really a wide variety of species in her career. She's looked at things like how chemicals influence monarch butterflies that migrate from southern Ontario all the way to Mexico. She's worked on how urbanization influences squirrels, gray squirrels around the city of Guelph. And she's been incredibly lucky because she's worked on lots of long-term studies. She's worked on sparrow savannas in the Bay of Fundy, and she's worked on red squirrels in the Yukon, and she's worked on gray jays in Algonquin Park. And those studies are incredibly valuable and special because the individuals have been the populations have been traced and tracked for decades, 50 years in some cases, on marked animals in the wild. And it allows her to have this really special lens on looking at stress in the wild. So I wanted to just give you guys a little bit of her history. She received her BSc from Queen's University in 2004. She then got her PhD in neuroscience from UBC in 2009, and she became a postdoc at the University of Guelph, an NSERC postdoc. And after 2 years, they loved her so much they made her faculty, and she's been there since then. She's won all kinds of awards in her career, many grants, many awards, many recognitions and prizes, NSERC, CFI. The University of Guelph has recognized her. I'm going to get this wrong, but their Research Excellence Award. She's received a Science Faculty Teaching Award, the Weston Family's Innovation Award, so lots and lots of awards over her career. She's published over 75 papers in some of the top journals in our field, including Science, Current Biology, Nature Communication. She's currently the editor, I think you're still the editor, maybe not, of Conservation.
I'm trying to
get rid of that. Okay, she's currently an editor of a very prestigious journal called Conservation Physiology. And I think because she looks at stress and the impacts of early stress on later adult behavior and neuroplasticity, through the lens of adaptation and through the lens of the environment instead of the normal lens that we look through, which is, biomedical, disease-oriented, I thought she'd be a really exciting and integrative speaker for our colloquium series. So I hope you guys will all help me in welcoming her. Thank you for coming, and we're really happy to have you. Thank you.
Oh my goodness. Thank you, Sigal, for the invitation and for everybody for taking time out of a very busy time of the semester. I know that it feels chaotic as classes wind down, exams wind up, defenses are going, field seasons are starting, and now I'm blushing immensely. That's such a generous introduction, and feeling a lot of pressure to tell you about some interesting things. I want to start with just acknowledging that a lot of the work that I'm going to talk about today is done in many different regions, mostly in Canada, but we have collaborators in the US as well. And so some of the traditional territories upon which we work are part of the communities of the Champagne and Asiatic communities in the Yukon. We work on trap lines of Agnes MacDonald and her family. On the other end of the boreal forest, we work in the Bay of Fundy, which is on the traditional territories of the Passamaquoddy, and then we do quite a bit of work too around Guelph in southern Ontario, and so are working on the lands of the Haudenosaunee and the Anishinaabe and all the members of those credit nations. And so I want to acknowledge that as biologists who roam broadly, that we try to respect Indigenous peoples across Turtle Island and Mother Earth, and also integrate their ways of knowing in the ways that we hope to conduct our research as well as our teaching practices. So I'm really grateful for those opportunities. This is a piece of art that Susan Matheson did. She's an artist locally out of Kitchener-Waterloo and did an internship with our lab and created this sort of summary figure for us, which highlights some of our study systems where you can see the savanna sparrows that you'll hear quite a bit about. And then there's gray squirrels running along the fence there. And the environments that are wrapped up in this image are sort of agricultural landscapes, as well as you can see the Toronto skyline in the background. So we're really interested right now in the Newman lab. They've labeled themselves as Newmanators. So the Newmanators are looking at landscape modification, agriculturalization, urbanization, and how that influences the physiology, behavior, and fitness of critters that live in those environments. And so we'll get to some of those stories near the end of the talk. What I'm going to do today is really just some storytelling about the systems that we're interested in and some of the findings that we've been able to glean from these study systems. We'll talk a little bit about birds and squirrels. And then if you have questions about some of the other study systems, I'm happy to chat about those too. But before we get started, I kind of want to put our research in a broader framework. Once we get the slides to move. This is just giving my sympathetic nervous system a chance to settle down. Where studying stress physiology in the wild, to me, is exciting but also a little bit unique, where we take the techniques and analyses that are traditional to lab systems and apply them to wild ecological systems. And we talk about how early life stress can shape physiology from both a prenatal and a postnatal perspective, and that we have different study systems that we feel best suit those questions. Sort of a classic August Crow principle where for any question that you have, there's an organism that's well suited to address those questions. And we know that the brain is very plastic, but also particularly sensitive to early life stress. And so that's why we're interested in that life history phase and the downstream long-term consequences that it can have on physiology, behavior, and fitness. But while working in a wide range of species, Sigal mentioned some of the study systems that we have ongoing in the lab in the past and in the present, it's clear that there's some well-entrenched paradigms, right? On the side of stress physiology, these are studies that are typically done in a lab under tightly controlled conditions where genetic and environmental variation is purposefully kept to a minimum. And those are super important. Most of you in this room are studying lab-based systems or systems in captivity. And what we want to do is to use that kind of design, but also incorporate variation that we would find in more natural environments. So when we think about the study of stress physiology and its impacts on things like immune function, hormone changes, neuronal integrity, for example, the stressors that we're applying in the lab are not necessarily ecologically relevant to what those animals would see in the wild. Grad students will be playing music to racks of rats or speaking loudly, unpredictable foot shocks, all things that are highly effective in eliciting a stress response, but not necessarily ecologically relevant. On the other hand of the equation, when we look at ecology and fitness, it's more broad scale, ecosystem level processes and patterns typically, where we're thinking about population dynamics, predator-prey relationships, the impacts of habitat change or climate, seasonality, migration, and they're all kind of at a bigger level. And what we really want to do through ecophysiological studies is blend those two fields. And so depending on my audience, I'm either an ecophysiologist or a physiological ecologist, a conservation physiologist. We like to mash up a bunch of words to try to describe what we do, but that hopefully will give you a sense of where our research program sits and how it integrates among those two fields. More specifically, we consider sort of stress physiology and the stress axis itself as a really critical proximate mechanism that links an organism to its environment. And so it's kind of like this master orchestra conductor that is just making sure that all of the different instruments are playing in harmony, but that one of the important insights from that analogy is that there are so many different instruments, both proximate and ultimate factors that can shape stress physiology or through the stress axis influence outcomes in a wide variety of systems and also at the level of the individual, and then scale up to populations. These are some of the critters that we focus on. So you'll see the juvenile phases on the left and the adults on the right, but savanna sparrows, red squirrels, and of course the ever iconic Canada jay, which was recently renamed from gray jays. And these are 3 long-term populations, all that have been studied for 40 years or more. 40 decades would be a lot. But it gives us an enormous amount of ecological knowledge about the individuals in these populations. And being able to track individuals over time and build pedigrees allows us to also look at heritability of some of these traits that we're looking at. So we conduct long-term observational studies, field experiments where we're manipulating some of these things, and also molecular analysis in the lab where we're often bringing things back, and students are working hard on those analyses, hormone, brain tissue, as well as genetic analysis as well. So those are kind of some of our long-term systems. And as I mentioned, we're really focused on the stress axis as one of those key mechanisms that link an organism to its environment. And because stress is a bit of a nebulous concept, I just want to kind of provide a general definition and give you some language about how we think about stress. There's quite a lot of debate, as I'm sure many of you are aware, about the definition of stress and how we can speak about it. Is it maladaptive? Is it an adaptive response? So on and so forth. But because we talk about stress and the outputs of the stress axis in this talk quite a bit, we'll just sort of talk about high level how we feel about it. So we measure glucocorticoids as one of the endpoints of HPA or stress axis activation. That's one of our most common endpoints that we measure. And I think that we can agree that a response to stress can be reliably characterized by an increase in circulating glucocorticoids. And glucocorticoids, for those that don't know, are sort of the output from the adrenals in response to hypothalamic pituitary adrenal axis activation. But generally, you can measure glucocorticoids in a variety of tissues and blood, fur, feathers, feces, as we do in all of those tissues, and I'll show you some data from those later. But circulating glucocorticoids are sort of bopping along at a baseline level until a stressor is encountered, in which case there's an activation of the HPA axis, and the stress response can be characterized by that increase in circulating glucocorticoids. And the concept of stress, like I said, can have a negative connotation, but I would argue that it's adaptive. These glucocorticoids are necessary to mobilize energy and to fuel that fight or flight response. And so being able to accurately characterize them in the field is really important for trying to understand the response to stress in a variety of species. Now, excitingly, the HPA axis, or the hypothalamic pituitary adrenal axis, is so powerful because it's highly conserved among terrestrial vertebrates, and indeed among all vertebrates, although fish are a little bit different. But generally, if we think about birds, which I've worked on quite a bit, you know, they have a hypothalamus, pituitary gland, adrenal cortex, just like mammals, primates included. And in response to a stressor, whether that be internal, whether it be real, perceived, external, predator threat, illness, acute sort of anxiety response, all activate the hypothalamic pituitary adrenal axis, or the HPA axis, and I'm just going to colloquially refer to that as the stress axis. And we can measure the glucocorticoid output in the blood or those other tissues that I mentioned. And the same is true in our squirrel systems, where that axis is highly conserved and it behaves the same way. And so we can ask common questions, and also the exciting thing is that we can perhaps start to draw translational conclusions that may apply to a wide range of vertebrates, including human health. People are really interested in how the urban environment could be inducing stress in individuals and how that can be characterized with some of these measurements too. So that's the framework of how we approach these questions. And I want to tell you about one study system that we focus on in the Bay of Fundy using savanna sparrows. Now, this is, as I mentioned, a long-term study, and we look at the impacts of the early life environment and how that carries over to adult stress physiology, as well as their behavior and fitness. Now, where do we study these little guys? They're ubiquitous little brown jobs. They nest in Walmart parking lots. There's not a lot that's, particularly special about them unless you're a bird nerd like I am and have a particular fondness for their beautiful little yellow eyebrow there. So they're quite lovely, but they're unremarkable. But what's remarkable about this population is that it's been studied, as I mentioned, for almost 40 years. And Kent Island is a tiny island in the Bay of Fundy, just off the coast of Grand Manan. And every year, the population is monitored, individuals are marked, and we follow the individuals that return year after year. In particular, there's a high level of natal philopatry in this population. And so island populations of songbirds have a higher return rate. And so then we can look at multi-generational impacts of some of these stressors that we're curious about that individuals experience throughout the course of their lifetime. It's a beautiful place to work. We take teams out every year, and we also connect. The island is Canadian soil, but is owned by Bowdoin College, which is a liberal arts college in Maine. And every year they have a cohort of undergraduates that come and live on the island at the research station and conduct research projects. And so we connect with them, we supervise some of those students, and our graduate students interface with them as well. We also like to employ child labor from time to time if it's available. They're really cheap. But like I said, it's beautiful. These are mist nets and how we catch birds in the wild. For those that are familiar with bats, it's kind of similar. And so we set up the mist nets and we'll catch the savanna sparrows during the breeding season, collect blood samples and tissue samples, as well as conduct our manipulations. And so I'm super grateful to the students that have worked on this project over the years. Being away from home for a long period of time is sometimes not easy, and these conditions can get a little bit challenging in terms of temperature, fog, wind all the time. Of course, I selected the photos where make it look idyllic and beautiful all the time. But we were curious about whether or not we could detect relationships between glucocorticoid levels and long-term behavior and fitness. And so we were asking the question was whether the HPA axis could be shaped by the early life environment. And the first thing that we had to do was characterize what those glucocorticoid levels look like in adults, whether or not there was variation that may be explained by the early life environment. And we were looking in both males and females, and at baseline and then after a period of acute restraint stress. So we can look at sort of chronic levels of glucocorticoids and then how individuals respond to an acute stressor. How reactive is that HPA axis? And what we found were significant differences both, of course, in the response to stress, but between males and females. And this is just sort of my opportunity to stand on my soapbox for a moment and highlight how important sex differences are. This community will appreciate that in spades, but I find that more broadly, people often will have a pet organism that they're studying, or males or females, perhaps because it's logistically more feasible or what have you. But not surprisingly, males and females are different. And how we approach the impact of environmental stress or landscape modification or drug exposure, we need to consider those underlying sex differences as well, because they can be quite important. And I'll give you just a teaser. I don't have the data yet because an undergraduate thesis student is just pulling it together. But these are total levels of glucocorticoids. And for anybody who's sort of in the weeds on the molecular angles of stress responses, we're also quantifying corticosterone binding globulin, and it responds to acute stress and opposite to this pattern. And so it could be masking the response to stress in some cases, or it could also be amplifying it. So really important to sort of pull apart some of those layers. But in general, adults have higher levels of circulating glucocorticoids at baseline and have a greater response to an acute restraint stress. But is that response shaped by the early life environment? And one aspect that's really important for wild birds is how much competition they have early in life. Now, how many siblings do you have in your natal nest? That means how much food mom and dad are bringing you, how much conflict there may be in the nest. For really low brood sizes, that might influence thermoregulation in a negative way if they're getting chilly. And so we know that the number of nestlings has an impact on behavior and fitness. But does it shape that stress physiology, and is the stress physiology a mediator of those impacts? So what we did was we compared, this is adult female corticosterone levels with the number of nestlings that she would have had in her natal nest when she was a youngster. And we've done some brood manipulations as well, where we've changed the number of nestlings in the brood and have seen similar results. At baseline, what you're seeing here for females is that the more nestlings that she had in the natal nest, so more competition perhaps, less resources, the higher the baseline levels of corticosterone are. However, that pattern disappears at the acute stress level. For males, we don't see a pattern at baseline. However, we do see a signal at the acute stress levels, where males that had more competition early in life seem to have a greater response to that acute restraint stress. And what this suggests to me is kind of an exciting sex difference in where early life stress may be programming the HPA axis. And so is it working more at the mineralocorticoid receptor level for females, the glucocorticoid receptor level for males? These are ongoing studies where we want to collect the brain tissue at this point and dig down into those molecular mechanisms. We've also manipulated glucocorticoid exposure early in life in this species. And so this is a study that was conducted by a master's student, Jesse Pakala, and his undergraduate research assistant, Catherine Smith. And what they did in savanna sparrow nestlings in the population is that they treated individuals with either peanut oil control or corticosterone twice a day for the first 7 days of life. And we did it twice a day because it has a short half-life, and we wanted it to be more of a repeated exposure type of situation. And then on day 7, when they were 7 days old, they measured their response, their baseline levels of glucocorticoids, and the response to acute stress. Then we let them mature, and we quantified the parent survival prior to migration. And what we found was that in 7-day-old nestlings, that there seemed to be a remodeling of the HPA axis, where that there was an elevation in baseline levels and then a dampened response. What this suggests to me is a dysregulation in the negative feedback of the HPA axis in response to that early stress exposure. And we see that manifesting later in life. So these are mature individuals who are getting ready for migration. And we see a reduction in apparent survival of those that were treated early in life with glucocorticoids, suggesting that there may be fitness consequences of that early life exposure. Now, one question that remains is, were these individuals dead or just not apparent? And so this is one of the challenges with field studies and tracking individuals and mark recapture studies is an alternative solution might be that they were so jacked up that they took off for migration early. And so we didn't detect them on the study site when we were still detecting those control individuals. And so we're following this up with more specific radio telemetry tracking, as well as long-term migration tracking with GPS backpacks. And so we can see whether or not they have survived and left early, or whether or not they have indeed had an increase in fatality prior to migration. Another aspect of this study that I find quite exciting is work that my PhD student, Haley Wilson, has been conducting, looking at female stress physiology and linkages to fitness. So we've gone from the nestling to sort of its juvenile state, pre-migratory state, and now when individuals return the following year to breed, does their early life environment that has shaped their physiology also ultimately impact their fitness? So Haley is interested in female fecundity, and fecundity in this case is a measure of an individual's reproductive success, how many babies they're producing over their year and their lifetime. And she was interested in linking female court with whether or not an individual mom would produce a second brood. So double-brooded individuals are those that will raise 2 full nests in a year. Often in wild populations of songbirds, female will lay a brood, and it will be depredated, and they'll have to do it again. And they'll keep trying and trying and trying. Levels of predation in this study system are traditionally quite high, mostly crows that are eating the eggs and the nestlings. But if a female is successful early on in the season, and she's able to raise that brood to independence, often individuals will nest again, doubling their reproductive success. What Haley did was create exclosures to protect the nests. And so everybody had a successful first brood without having to re-nest due to predation. And then she looked at, for those that decided to double brood, did corticosterone levels predict that probability of double brooding and increase their fitness through reproductive success? And the answer was, interestingly, yes to a degree. So we see that individuals that double brooded, that had lower baseline corticosterone, those were the ones that were more likely to double brood. But we don't see the pattern after those 30-minute levels. So again, here's the baseline corticosterone levels that seem to be physiologically and behaviorally relevant for females. We don't have similar measures for this in males because we don't have as much intuition and insight as to how males influence the decision for females to double brood. Males will take on as many females as they can. You know, there's a high level of promiscuity in this system. So this was a study that was focusing only on females, looking at impacts of corticosterone on fitness. Now there's a lot of sort of nitty-gritty proximate elements to these studies, and I think we're getting at some of the pieces that look at the impacts of the early life environment on shaping stress physiology and how that can carry over into adulthood. And what I'm really excited to do is to think about this more broadly in a conservation physiology perspective. Because we know that since 1970, that there's been a drastic decline across bird species on the continent. The estimate is that since 1970, about 3 billion birds have been lost. And the reasons are multifaceted. We've got landscape modification, climate change, exposure to toxins, all sorts of different stressors that are facing these types of organisms. But what's noteworthy from this particular figure is that it's the grassland birds that are experiencing the most significant decline in North America. Grasslands are one of the most threatened ecosystems on the continent and indeed across the planet. And so I think that we can use some of these insights from savanna sparrows, which are grassland breeding birds, to try to understand the conservation implications for these massive declines. Because I really do think that birds are the canary in the coal mine in this case in terms of trying to anticipate and understand long-term ecological collapse. And so I think that we're really at a precipice where we need to understand both the physiology and the ecology of these organisms. And this is a neat graphic that came from that same study that sort of demonstrates how dire it is for grassland songbirds, and that we can use these little guys who are not threatened, endangered, or seemingly experiencing declines to try to get at some of those insights. And so can we use our understanding of early life stress through these physiological measures to make these meaningful contributions for applied conservation? And I want to take just a minute and give a nod to some work that we're doing more locally, also with savanna sparrows, and in collaboration with Alice Farms. Alice Canada is this great organization. Has anybody heard of it in this area? It's a couple of nods. Awesome. It's a real grassroots, farmer-led organization that supports individuals in converting marginal farmland into restored habitat, riparian zones, tallgrass prairie, reforestation, in order to provide ecosystem services that are of benefit, of course, to the wildlife that are using these agricultural spaces, and also more broadly to Canadians as we think about sustainable agriculture and how that interfaces with stress and wildlife. And so we've been asking questions in collaboration with farmers that are using these Alice practices. Here's some of the field teams that have been working in these agricultural areas, and we compare individuals and their stress responses as well as neurophysiology in farms that have restored farmland as well as conventional farms, and comparing that to conservation areas. Trying to understand how these sustainable agricultural practices may also enhance the fitness of wildlife, in particular avian wildlife. And so just to compare these two systems, we can use the insights that we glean from Kent Island and we can apply them more locally to agricultural lands. And what I'm really excited about is some experimental manipulations where we can... Is that me? Okay. I mean, I knew it wasn't me, but where we can treat individuals with agricultural pesticides in our Kent Island population, where they've never had to interact with those kinds of stressors before, and look at how that impacts their stress physiology and brain development, as well as their fitness, as well as look at how variation in some of these environmental practices can impact birds that are on the land. And so using this one species, I think we can get some really exciting insights on a number of those different questions. So I want to switch gears a little bit. That's sort of some of the work that we're doing from a postnatal early life perspective using a long-term population. And I want to switch gears now and talk about a little bit of the work we've been doing on prenatal stress, shaping offspring physiology from a prenatal perspective using a different long-term study. So this is the Kluane Red Squirrel Project. These are North American red squirrels. And we study these in the field as well. And this was a study that was inspired sort of by the framework that's attributed to Michael Meany, but is, of course, really quite relevant to a broad range of researchers, where generally it's known that early life stress can shape a mother's phenotype, and mom's stress physiology has a relationship to her maternal behavior. And this sets up, it's a spectrum, but on either end of the spectrum, we can have moms who provide high levels of maternal care and moms who provide low levels of maternal care. And so what you can see in these photos is kind of a comparison where this mom is providing lots of licking and grooming behavior, keeping the pups all nice and cozy in the nest. If one wanders away, the pup retrieval time is quite short. And that's in comparison to a mom that provides lower levels of maternal care. So you can see there's very little interaction or licking and grooming. Pups are spread out. If one goes awry, then pup retrieval times tend to be longer. And so these two types of maternal phenotypes then lead to differences in the F1 offspring, where we have offspring born to moms who provide high levels of maternal care, who tend to have, nice perky ears and happy tails, but also higher levels of glucocorticoid receptor expression in the brain. They have lower levels of peak cortisol. Their HPA axis is nice, finely tuned. It shuts down very quickly. That negative feedback is working well. They tend to express lower levels of anxiety behavior, and they themselves provide higher levels of maternal behavior to their offspring. So through epigenetic effects, we can see this persist. And that's in contrast to pups that are generally born to moms that provide lower levels of maternal care and behavior. So what we wanted to do is take this paradigm into the field. And as I mentioned, we did so with this critter, the North American red squirrel. And so this is sort of a prenatal perspective on the potential for the early life environment to shape offspring physiology and fitness. Now, the Kluane Red Squirrel Project is part of the larger Kluane project that was started by Charlie Krebs and colleagues many moon ago. But the Red Squirrel Project has, like Kent Island, been monitored since 1987. Coincidentally, they both started in the same year. And we've been collecting physiological data since 2009. Like Kent Island savanna sparrows, there's a high level of natal philopatry, even higher than the sparrows, where individuals, if they survive, will recruit into the population 400 meters or less from where they were born. And so here we have incredibly rich pedigree data from the population for decades. And so that gives us a lot of power to ask a range of interesting questions. And field work here is also fantastic. Starts in February when it's -40. The field station itself is very rustic, but it also turns into sort of this beautiful habitat that we can enjoy, and the teams work really hard. So I'm also grateful to all of the students that go up into this adventure land year after year. It's quite a special place. But the question was, does prenatal stress influence the physiology and fitness of offspring? And so what we would do is every year all of the squirrels are individually marked so that we can recite them without having to catch them. But we would trap them regularly to monitor their states of pregnancy. And when we identified a mom who was in the equivalent of her 3rd trimester, we exposed her to a stressor. We know in this population that population density is a really potent stress for these individuals, that individuals living under higher density have higher levels of stress markers. But we know that from some of the individuals that are in high density because they've also received supplemental food. So part of this study, long-term study, was really neat where they would supplement the amount of food that the squirrels had, and over years the population would increase because there were more resources. In those individuals, in those higher density areas, they had higher levels of stress hormones. But we wanted to take food out of the equation and just have it as about population density being a stressor during pregnancy. And so in squirrels who we identified as pregnant, we surrounded them with playbacks, playing squirrel calls. And so they perceived that they were living in an area of higher density, where we know then that that this did in fact increase their levels of fecal glucocorticoids. So we're measuring cortisol in the feces in this case, which is quite easy to obtain if you can trap the squirrel. And we saw that moms who were exposed to this treatment of perceived high density had significantly elevated fecal glucocorticoid levels. So we confirmed that that manipulation impacted mom, but what we wanted to know is what was the long-term impact on the offspring. And so after individuals were born, they're measured on the day of birth. We collect their birth weight, some genetic data. They're too little at this point to get a decent blood sample for any hormone measurements, but how fast they grow, their growth rate is directly tied to their long-term fitness. And then again, 25 days later, we would return to the nest, measure them again, and here we would collect some blood samples and permanently mark the individuals. What we know is that before we did our experiment, that individuals under high density, high food conditions tended to grow faster. The faster you grow as a pup, the faster you get out of the nest, the more likely you are to secure your own territory and survive the winter and live to breed the following year. If you do not secure a territory, the likelihood of overwinter survival is almost none. And so your growth rate is tightly connected to your lifetime fitness. So here we have individual squirrels, and what you're seeing with this decrease is that litter size impacts the growth rate. So the more competition there is, the slower individuals grow. You can imagine that if mom has to nurse 6 pups versus only 3 pups, then those individuals have fewer resources to go around. Then we compared what happens with our perceived high-density treatment in the absence of supplemental food, and we find that those individuals grew just as fast as those that were born to the moms with supplemental food, suggesting that that prenatal stress of perceived high density was enough to influence the growth rate and get those pups out of the nest faster, especially under high density conditions, because competition for those territories is going to be high. So we know that that early life stress is shaping the development of these individuals. Now we wanted to dig down a little bit deeper and look at the underlying physiological mechanisms. So what we did was we tested their HPA axis response in the pups that were born either to the control moms or the moms that were in the playback situation using a dexamethasone ACTH test. And this is a pretty standard test where dexamethasone is a synthetic glucocorticoid, so it activates the negative feedback and should shut the stress response down. So in a typical HPA axis response, we would expect that in response to dexamethasone, there would be a reduction in circulating glucocorticoids. Then we can inject ACTH, which should stimulate the adrenals to produce glucocorticoids, and we can measure the duration of that response, akin to what would happen in response to an acute stressor. In an individual that might have been born under stressful conditions or in exposure to prenatal stress, we might expect that the HPA axis becomes dysregulated. And so what we might see is an elevated level of circulating baseline glucocorticoids, a reduction in the response to dexamethasone. So dexamethasone resistance can be a sign of a dysregulated HPA axis. And then some sort of dysregulated response, whether or not there's a lower sort of peak or elevated period of time where glucocorticoids are being released, that there's some sort of dysregulation in the ability to turn the stress response off. And so we looked at this response in the adult offspring that were born to these stressed moms. And we do find some evidence that individuals that were born under prenatal stress conditions, so the playback moms, have a reduction in their ability to respond to ACTH, and overall seem to have a longer period of glucocorticoid production. So we actually measured again after 3 hours, and we find that this line continues out further, suggesting that there's more glucocorticoid production, that the HPA axis is on for longer periods of time. Nonetheless, it seems to also have dampened the amplitude of this response. And so that there is dysregulation in the HPA axis. How that impacts fitness is the next question. And so are these animals perhaps more adapted to high stress conditions so that they're more resilient? Or is this dysregulation actually having fitness consequences whereby perhaps they are not responding to their environment in a way that is quite as appropriate? So those studies are ongoing. But from these individuals, what we did then was we collected brain tissue, and master's student Kevin Morey conducted some qPCR for glucocorticoid receptor expression in brain regions that are associated with the HPA axis. And we're also working on some DNA methylation analysis for epigenetic signatures in these offspring. So this is the strategy that Kevin used for excising brain tissue. These are micropunches that are being collected from brain sections as the brain is being sectioned. And what he found was that there are seemingly sex-specific effects in terms of how individuals responded or were impacted by the early life stress exposure. So he looked at mRNA expression for glucocorticoid receptors across 3 regions of the brain, and I'll just orient you to this figure, where we have control and prenatal stress in light and dark gray, and then the hashed marks are the females, and those that are unhashed are the males. And so across the brain, we see some really interesting patterns that are perhaps a little bit unexpected. There's a ton of variation because our sample sizes are quite small, but some suggestion that we see that there are sex-specific effects, particularly in the pituitary. And so we want to follow that up with some more sophisticated analysis in future studies. But I think that this is really exciting evidence that there is neuronal programming happening in response to the prenatal stress that carries over into adulthood. And we can imagine that that would impact individuals' responses to stress as an adult themselves, and perhaps have epigenetic effects for generations into the future. We have some other neat, more behavioral studies going on in the red squirrels as well about how they can transmit sort of acoustically their own physiological stress state to their neighbors, and whether or not that impacts the neighbor's stress physiology. But I'm happy to chat about those afterwards as well. What I want to end with is one last sort of story that we're working on more contemporarily, where we're also interested in whether or not environmental stress through vis-a-vis urbanization can impact individual physiology and also be transmitted to offspring. So we're asking questions along the lines of, does city life make a difference for individual stress physiology, and in particular the gut-brain axis? We're getting very excited about microbiome in the bidirectional connections between the stress axis and the gut microbiome. And for this, we're using gray squirrels, another ubiquitous critter that's colonized almost every continent across the earth, sometimes to great conservation concern. But they're very tractable for comparing individuals in urban and more natural non-urban environments. And we can radio color them and follow them for long periods of time, as well as access their nests and get connected with their pups as well. So here's some of the field team that's leading some of these studies. Alana Grant, I'll give you sort of a teaser at the very end of what she's looking at, but it's also, those data are going to be hot off the presses very soon, I hope. But for those that are not familiar, just kind of broad, high level, that we all harbor these multicellular communities known as microbiomes. The gut one probably gets the most attention, but they are on every surface that's exposed to the environment. And it's really important to understand both the interactions between the microbiome writ large, but also who are the players in that microbiome. And as we sort of develop metabolomics and those studies become more sophisticated, we're getting some really exciting data that's specific about what kind of gut bugs are in that microbiome and how those are influenced by stress. But generally, host physiology is thought to shape the microbiome. The environment, we know, impacts host physiology. But now what we're learning is how that microbiome can feed back and influence individual physiology. So that's kind of the broad framework that we're working in for the Gray Squirrel Project, and also incorporating some of those really key factors of an individual's life history: sex, reproductive condition, age, season, etc. All of those things that we know from our previous work have really important impacts. And how we consider this is sort of, again, that HPA is that physiological intermediary that connects the organism to its environment. And we know that cortisol, through activation of the HPA axis, can influence such a range of endpoints. Again, coming back to that, to the analogy where it is sort of this master conductor of a really sophisticated orchestra. And these are just a few of those important behavioral and physiological endpoints that we're considering and that we're analyzing. So this study that I'm telling you about is occurring in a series of paired locations across Southern Ontario. I'll just focus here on one pair where we're using the University of Guelph campus as our urban site and the Rare Charitable Research Reserve, which is in Cambridge, as the naturalized site, and doing some comparisons, both behavioral and physiological, on these critters. And so it's quite straightforward to trap the squirrels, and we collect morphological measurements, blood, fecal, and hair measurements. And these are nice because they give us a variation in time frame of what they reflect, sort of that stress physiology, where blood is quite acute and immediate on the order of minutes. Fecal samples give us more of a hour to day-long time frame of what the circulating glucocorticoids have looked like. And then they get deposited in hair during molt. And so this gives us more of a month to year time frame about how we can backcast the stress physiology of an individual. And we have some measures that suggest there is environmental variation among these different endpoints. So for blood, feces, and hair, we see differences in the urban versus naturalized environment, not always in the same direction based on what time frame you're looking at. So this is kind of an interesting consideration too, is that acute stress and chronic stress can have very different impacts on an individual's physiology and how it shapes the microbiome. Another important thing that we needed to establish early on was, does the microbiome community structure look different in our different environments? And indeed, these data suggest that it do, that the diversity of the different types of microbiota that are in the microbiome are distinct in individuals that live on campus in an urban environment and in a more naturalized forest environment. And if we take just a quick look at what some of those communities look like, here we have squirrels. So each column is an individual squirrel, and then the different colors are groups of the microbiota that fall into these different groups. And you can see from forest to urban, there's a lot of variation, but also some consistent patterns that are coming out, in particular individual microbiota that are important for different aspects of a squirrel's life history and diet. I'm going to focus on just a couple here. We have Akkermansiae and Lachnospiraceae, and we can see that there are relationships between hair cortisol and levels of those critters in the microbiome. And this is important because we know that these two critters have very different roles, right? Lactospiraceae are known to be fiber digesters, and we see that they tend to increase in response to stress, whereas Akkermansiae are known to be mucin digesters, and those decrease in response to increasing hair cortisol. And so this is important because it raises the question whether or not chronic HPA activation can influence the regulation of the microbiome. And so we're getting now into some of these sophisticated connections between the gut-brain axis, looking at how an individual's stress can influence the microbiome and reshape it such that it would change the function of the microbiome and had feed-forward impacts on future behavior. And that's what Alana is kind of interested in, trying to understand. Do offspring born to urban moms who have higher glucocorticoids have lower rates of neurogenesis that are associated with shifts in the microbiota? There's another group of gut bugs that are influencing directly neurogenesis, and so we're also quantifying those with the hopes of, in the future, being able to manipulate the microbiome through both corticosteroid treatments in the wild, but also by inoculating individuals with different microbiota. So those are a few of the stories that we've been focusing on that I hope give you a broad view of how we are looking at the early life environment, environmental stress, and how it shapes individuals' physiology, behavior, and fitness. And with that, I'll thank the critters that have participated in the studies over the years without their consent. And I'm really grateful to you for taking time, again, out of your day, for Sigal, for the invitation. Some of the funders that have funded the work either directly or through scholarships, and then just my information if anybody else is excited about these sorts of things. We are always looking for great students to join the Newmanator team, so feel free to reach out. Thanks, everybody.
Okay, well, we have a reception afterwards, but I'm sure there are questions from the audience, and we have plenty of time for those. So does anyone have any questions? I'll let you just field your own questions.
Okay, certain. Paul.
Just a quick context question. How long do reds roll live?
In the wild, on average, if they survive their first year, 4 years.
Thank you.
Alex.
Thank you for a great talk, Amy. I'm less well-versed in this field, so I was wondering how you account for, like, you talked about acute stress, but I'm sure, like, the catch and release portion of what you do, at least in the field, is quite a big stressor for these animals. So I was wondering how you account for that, or if you account for that.
Yeah, it's interesting. It depends on the study system. So and sometimes we're trying to leverage the fact that the catch is the stressor itself and what activates the stress response. But we do try to take care and monitor individuals after they've been released because we can keep eyes on them and be careful not to catch individuals too frequently in order to continually activate, the acute stress response. But otherwise, it's sort of one of the things that, like, through a welfare perspective, you try to keep an eye on in terms of the animals, and if somebody seems a little bit amped up, then we don't catch them, or if there's a sensitive period too, so if a nest has just hatched, for example, then probably we'll avoid trying to catch the adult female on that day, just so that you're not pulling her off her nestlings early in their life too.
Thank you for the great talk. I was wondering, related to the red squirrel, the prenatal effects of perceived stress or density. Wondering if you think it's potentially behavioral effects of the dam once they've given birth. So there's a prenatal part, but then also the postnatal part of the care that is provided by the dam. What combination do you think is driving the effect?
It's a bit of a black box, but we're trying to get at it. So the following year, we didn't do a playback, but what we did was we fed mom glucocorticoid-laced peanut butter just after birth. with the assumption that that would change her behavior, but we don't have a way of actually getting into the nest because they are just like a mouse nest, really, a fluff ball. So we can't measure her actual behavior. But based on the previous work that's been done in the lab, I think that licking and grooming and provisioning is really probably what's contributing to those growth rates. To do like a cross-foster? Yeah, we've done that in wild mice, and so just not in the framework of the playback experiment. And so that would be sort of like the unicorn at this point to do that in the wild, and something that for the right student, because it is so much work to find the nests that are synchronous. And so there's a lot of variation in birth dates. And so if we're not switching individuals that are exactly the same age, then it becomes really difficult. But I would love to. Yeah. Back. Oh, thank you.
For the Savannah Sparrow project, you mentioned the next step might be applying some pesticides to the nest and to see their stress response. But I was wondering, have you looked at if there is a correlation between like the sites that you're already monitoring the individuals, like their pesticide levels and their stress levels? Because I'm assuming if you're catching them, it's really easy to just take some clean oil sample and then run some analysis to see if there's a difference in the pesticide levels, and of course there's individual differences, and whether if you see like there's a positive correlation.
We haven't been able to do that yet. Preen oil for, oops, sorry, I keep, for anybody online. Preen oil for these little guys is hard to extract enough. But what I do have in the sort of hopper waiting for the analyses now are fecal sacs from the nestlings. And so we've been analyzing those for pesticide residues with the assumption that mom and dad are catching insects on the environment that are going to be treated with pesticides, agricultural chemicals, feeding them to the offspring, and whether or not we can pick up that signal in the fecal sacs. Yeah, good idea.
I have kind of a follow-up question. You showed at the very beginning, I think it was with the Savannah sparrows, that you have a very different sex response depending on whether you're looking at acute or chronic stress. And you saw and how many siblings you had, how much competition you had when you were growing up has this very different effect depending on your sex and what kind of stressor you're applying, chronic or acute. Has that been found in other species, and why are we seeing those stress differences? Why do we see it in males in the chronic, I guess it was acute, but what's going on? Why would you get those sex differences, and do we see that in other species, even in ourselves?
We do. There are sex differences, and that get teased apart from chronic versus acute responses. The meerkat population is one example where there's evidence for sex differences and time frame differences. I think that it's because males and females have such different roles to play, and that the impact of being responsive to stress or having an adaptive response to stress at baseline versus sort of your activated state is very different for males and females, where if you have an individual that's involved in territory defense, for example, versus an individual that's involved in offspring rearing, that those types of behaviors are so different and rely on different responsivity at those different levels. So that's kind of my broad scale guess, although we haven't done any manipulations to really drill down into that. But I think it's so fascinating. And then, to think about species where you have biparental care versus uniparental care, territorial versus social. I think there's so many different layers to it that I think are exciting. Back?
I think you mentioned you have some data on acoustic social signaling of stress in squirrels. Could you tell us more about that?
Yeah, so this is work from a PhD student who was interested in whether an individual's glucocorticoid levels, this is in the red squirrel system, who have pretty structured territorial interactions, whether an individual's kind of glucocorticoid levels could be transmitted acoustically. So would we pick up signals of stress in the voices of the squirrels? So you think about songbirds, and song production is influenced by stress, but we tend to think less of this in terms of mammals that have, to us, which sound like very uniform vocalizations. But what we've detected is that vocalizations change with different stress states. And so then we're curious, if you hear a neighbor who's giving you a stress signal through their vocalizations, does that influence your stress, and does that transmit through the network? And we're finding evidence that it does. And is that a communication strategy for, there's a predator in the area kind of thing, alert everybody, or do individuals want to conceal their stress state if it's an internal physiological thing that might make them more vulnerable? So we're trying to tease some of those things out.
Hi, that was really wonderful. Thank you for sharing all of that. I was wondering if you could talk about the red squirrel data where I think it was the offspring, you inject something that makes the squirrels have that negative feedback loop, and then inject something that has them stressed acutely.
Yep.
And then the ones that seem to be dysregulated they have a higher baseline and also respond for a longer time. I was wondering if that affects their behavior, and if it means that they couldn't really respond to multiple acute triggers of stress?
There's some evidence from different systems that suggests once you're activated that it does get harder to be more dynamic. But people tend to equate it to anxiety kind of activation, where you're in a constant state of being on alert. So does that eventually exhaust your ability to respond to future stresses? Quite likely, yes. We didn't test that behaviorally here, but what we do see is that in squirrels, both the red squirrels and the gray squirrels, that individuals with higher levels of glucocorticoids or with evidence of that dysregulated negative feedback do express your kind of classic types of stereotypic anxiety-like behaviors in an open field trial. And so we can put them in a trial box in the field and record their behavior, and they are behaving quite differently, where they're sort of trying to, spin around the outside the box, dig their way out. Some of them freeze, and then we can also expose them to stressors in the box, like predator shadows and things like that, and look at their response.
Maybe one last question, if anyone has one. We have one.
I have a question about the DNA methylation data and the work on the glucocorticoid disorder, the receptor gene, and I was wondering, you showed the mRNA expression data, so you could see the expression is different, and I was wondering, where were you noticing the differentially methylated regions between those two?
Stay tuned.
Oh, okay. I will.
If I had the data, I would show you, because it's something that we're pretty excited about. I mean, if I had to guess, it's going to line up in the pituitary, but we will see. Yeah, sorry, that was kind of an anticlimactic final response.
Okay, everybody, there's a wonderful, I'm sure, reception, because our committee's done such a